Hi everyone, I'm Nadir. And first off, I'd like to say the summer was really fun for me. I have developed an extreme appreciation for neurobiology. It's been a real adventure with lots of ups and even more downs, but we're on an upward track. And I'd like to share my adventure with you guys, so I'd like to make this enjoyable as possible. So starting off, what does this title mean? Characterizing D1R expressing neurons in the medial prefrontal cortex. Well, D1R expressing neurons are dopamine, D1 receptor expressing neurons. So dopamine has two families of receptors, D1 and D2, and we'll get more into that. But last Friday, I ran across a fortune cookie at the bus stop, and it was folded up, and I unfolded it, and it said, ears hear and eyes see. What then does mind do? And I thought, that's exactly what I'm looking for right now. Where is the mind? Well, I would like to say part of it is in the prefrontal cortex. You can see the prefrontal cortex, way up there, receives a lot of input. You have your visual input coming from the occipital lobe. You have other sensory inputs, auditory, spatial inputs. So the prefrontal cortex is kind of like a thinking center of the brain, involved in a lot of executive and thought functions, whereas other parts of the brain, maybe the back of your head is involved in visual, or the top of your head in somatosensation. As you can see, we're interested in the medial prefrontal cortex, which has a lot of projections from the hippocampal formation, which is involved in memory storage, the amygdala, which is involved in fear and rewards, temporal visual association areas. So a lot of sensing is going into the prefrontal cortex. And we're looking at the prefrontal cortex in terms of rewards and learning. So maybe if you got a stimulus saying, oh, here's a reward, the visual or medial prefrontal cortex could go to the hippocampus and say, hey, is this reward, do we know about this? Then the hippocampus could say, yeah, we remember it. Hey, amygdala, is this a bad reward? Should we be afraid of it? No. Okay, let's go to the dorsolateral prefrontal cortex, go to motor structures, and retrieve this reward. So, we're looking at D1 receptors. That means dopamine has to bind to this receptor. That's dopamine, and dopamine is a neuromodulator. What that means is that it modulates the response inside of the neuron. It's not just simple excitation or inhibition. Dopamine is involved in neuroplasticity in that when it binds, it changes the shape of that neuron and its ability to respond to subsequent stimuli. So it's akin to a sort of learning signal involved in plasticity, where it can strengthen preferred inputs. Say you get a chocolate bar, and then you know where that chocolate bar is. Dopamine would say, hey, we know exactly where to go for that chocolate bar. And it can weaken non-preferred inputs. Like if you burn your hand on a stove, dopamine would create a neural circuit saying, don't do that again. Remember that that's a bad response. So now we have two different dopamine receptors, D1 and D2 receptors. And there's a problem, because these receptors are G-protein coupled receptors. They initiate secondary messenger cascades involving a lot of responses in a cell. D1 receptors are excitatory. So you see they increase cyclic AMP expression. It could lead this cell to propagate a signal onto later cells. D2 receptors are inhibitory. They decrease cyclic AMP expression. So dopamine binding to a D2 receptor would stop the cell from propagating a signal. And how do we develop a coherent response is the question, especially when you have D1 dimers, D2 dimers, and D1 and D2 dimers just increasing calcium in the brain, or calcium in the cell, which calcium is involved in a lot of other neuroplasticity responses. So we're wondering, you have dopamine released in the prefrontal cortex when you sense something. Dopamine binds to excitatory receptors, inhibitory receptors. Some of these cells have both receptors on them, some have only one or the other. How do you generate a coherent response? So, this is an example of the D2 receptor, its cascade. I'm amazed when I see this because I think, wow, this one molecule can do so much with a cell. So I think we're on the very tip of a huge iceberg about learning processes. How are we looking at this? We use a mouse model with a discriminatory learning task. Discriminatory in that you have a positive stimulus and a neutral stimulus. If the mouse licks a rod for water after a positive stimulus, the mouse gets water. If it licks the rod after a neutral stimulus, nothing happens. We'll go into the stimuli later. And we're also using optogenetics and electrophysiology recordings. We're using transgenic mice, so This summer I was working with D1 receptor Cre mice, and like we saw in earlier presentations, Cre allows us to selectively express certain proteins. So we can record at the individual neuron level to see firing patterns in single neurons and correlate those neurons with behavior. How does optogenetics work? Long story short, we found a protein, channelrhodopsin, in light-sensitive algae. Channelrhodopsin is a light-sensitive protein that excites a cell. Somehow, through a long, complicated process, we're able to solely express channelrhodopsin in one type of cell. That would be the D1 receptor cell. Our cells expressing the D1 receptor express channelrhodopsin. So now you can cause neurons to fire just by flashing blue light into the brain. How do you get blue light in there? You put an optic fiber inside a mouse's skull. And then when you shine light, you get neuron firing. So, exactly what does this optrode look like? This is what I had to build over the summer. There's an optic fiber and electrodes on that end, and the wires going to a connector over there. When I first started, it was pretty tough. I didn't do so well. And I got better at super gluing the wires together. These are 20 microns in diameter. And then finally, I got it. And this took me maybe a better part of 50 hours to finally, it's like surgery on these wires. So what is this exactly? There's an optic fiber here, and it's surrounded by a bunch of electrodes. So we shine light through the optic fiber, and we're able to record what neuron activity through the electrodes. And at the other end, you have empty wire ends, and you connect those to connector. And the finished product looks like this, where you have your part that goes in the brain. This part goes out to a preamplifier and an amplifier where we can record signal. So learning task. We can't record the mice in the Northwest Animal Facility because of whatever regulations, so I may do. The learning task is we have a mouse, and there's a rod that it would lick for water. And there's a light, which is our stimulus. The mouse, in order to stop it from moving, we head fix it and put it inside a capsule so that it restricts movement. Why do we have to restrict movement? Because movement generates a lot of electrical activity in the brain, and we have to be able to record individual neurons, which produce really small levels of electrical response. So, we also put the electrode in the brain, and when the mouse licks the rod, if it's during the right stimulus, the mouse gets water. If it licks the rod at the wrong time, it doesn't get any water. So eventually it learns to associate positive stimulus with water. And it usually always gets water. So, what exactly is our behavioral trial like? This trial runs over the course of an hour. And for 1 second, we shine a stimulus, a light. CS minus is conditioned stimulus minus, in that there's just a long pulse of light. CS plus is 3 dashes of light. And if the mouse licks the rod after the CS plus, it gets a reward. And there's a 1 second window in which it can lick. And 5 seconds later, we shine light. And we shine the laser to stimulate the neurons. And because this isn't during the actual trial, we can record baseline activity. We can see, hey, these neurons are spiking. We're recording actual neurons, D1 receptor neurons. So the schedule for this is first we do a virus injection just to put in our channelrhodopsin in the mice. We habituate the mice to drink water from the setup because mice don't just drink water from rods, especially when confined. Then we do associative habituation. We start shining a light, and the mouse licks when it sees the light. And then we put in the optrode, the optic fiber and electrodes, and we start the discrimination task. And this is the big money when we can observe how the mice react to positive and neutral stimuli and record their neuron behavior. Then we can get our results, correlate behavioral patterns with neuron firing. I was so excited when I saw this, because this is, these are 2 neurons. One is the red, that's the shape of that spike from that neuron. The green is over here. What we correlated with is this 100 point is at the CS plus, right when the light flashes for the positive stimulus. And we see, hey, this red neuron has a huge response. On the y-axis is the fraction of responses over all CS pluses. The red neuron almost only fires during the CS plus. The green neuron has a lot of low-level baseline activity, but doesn't show any specificity for this response. So we can say, hey, in the prefrontal cortex, we have some cells that are specifically responsive to positive stimuli. And another way we can differentiate between cells is looking at how they respond to light stimulation. One thing we realized after looking at this figure is that these are all different cells and how they respond to light stimulation. Over here, we can see that this cell response to light stimulation is 20 hertz per 0.5 seconds, so the light flashes 10 times over the course of half a second. We can see later on, the cell responds more and more, which means it's probably indirectly stimulated by light. Whereas the red neuron, from the get-go, fires every single stimulation. The blue neuron, when you correlate it with the red, it's not as strong of a firing, so we can say that the red neuron is probably a D1 receptor neuron. But we realize that for the future, we'll have to lower the wattage of our laser so we don't get these type of responses where we're indirectly activating neurons. On the third day of recording, our optrode lost signal. This could be due to a cut in the reference wire when we have to, or the cut in the ground wire, and we weren't able to gather data for more behavior trials. So for the future, we'll just have to run more mice and get more data. But we saw that some neurons respond to CS plus stimuli. What about licking? When do neurons fire with licking? When do they fire with CS minus stimuli? When do they fire in response to rewards? These are all questions we're looking to answer. So these putative D1 receptor cells, in order to confirm that they are D1 receptor cells, we'll have to revise our optogenetic protocol and do more precise laser stimulation to record the individual neurons. The questions we're left with today is, what is the rationale for this complex circuitry? What role do D1 receptors and D2 receptors play in cells, especially when they have antagonizing responses? For the future, we just need more mice and more data. We saw cells that responded to CS+, we just need to see more cells that respond to different stimuli. Maybe, it's a hypothesis, maybe D1 receptor expressing cells fire with a positive stimulus, maybe D2 receptor expressing cells fire with a negative stimulus. We don't know, we just have to find out. So we have to record D1 receptor expressing neurons, record D2 receptor expressing neurons, and correlate their neuronal action with behavior. What's the difference in binding from the D1 receptor to the D2 receptor? How does it relate to our thinking processes? And lastly, we'll have to do some histology, some anatomical studies. Where are these neurons going to in the cortex? Where do they project to? And what goes in there that could modulate these inputs? Lastly, I'd like to acknowledge the CERT program, Rose Hills for making this all possible, the Dan and Poo Labs for sponsoring my research, my postdoc Andre, Nathan for being our CERT mentor, Dr. Carroll for supervising this whole fellowship, Ken, Michael, and Evie, the undergrads who work in my lab, who have supported me all the way through. Thank you.